Hi, in this lecture we're going to talk about renal physiology and the role that your kidneys play in fluid and electrolyte balances. If you look at your urinary system is composed of your two kidneys, the two ureters that lead from your kidneys to your bladder, the bladder, and the urethra. For the most part in this lecture we're going to focus on the kidneys because the kidneys are what are doing most of the work. The bladder is basically to store the urine and then to excrete it. The kidneys' function is to make the urine. And what the kidneys do is it basically filters your plasma or your blood. And it's going to eliminate any unwanted components that are found in the plasma, such as waste products, and produce urine, and you'll excrete that urine. But what the kidneys also do is they conserve any materials that the body needs. So what it does is it excretes unwanted waste products while conserving material you want to keep in the blood. So let's look at some of the many functions of the kidneys. One is to excrete nitrogenous waste. So nitrogenous refers to anything that's nitrogen containing. And a lot of these waste products come from metabolism, such as urea, uric acid, and creatinine. Another one is ammonia. These products can be toxic to the cells, even though they're a product of metabolism, so they need to be excreted. The other function of the kidneys is the kidneys play a role in regulating blood volumes. The way it does that is it controls water reabsorption, so the kidneys can either excrete more water to get rid of extra water to lower your blood volume, or it can reabsorb water to maintain your blood volume. The other thing it does is, remember, the kidneys release the hormone erythropoietin in response to low oxygen levels. This causes red blood cell production, so it could kind of affect the viscosity of the blood also by producing more red blood cells. The kidneys play a role in blood pressure. We've already learned a little bit about this, and we're going to talk about it some more, and you're really going to start to see how many of the systems work together to control and maintain homeostasis. So the kidneys regulate blood pressure by releasing renin, and you've learned that, remember, renin triggers the renin-angiotensin-aldosterone system, which causes vasoconstriction, which will increase blood pressure, and aldosterone secretion, which leads to sodium reabsorption followed by water reabsorption, which helps you maintain blood volume, therefore blood pressure. The other thing is it regulates all the chemical composition of the blood, and the way it does this is it regulates which ions are reabsorbed and excreted and other solutes, so it also affects osmolarity. It helps stabilize pH because the kidneys can either excrete acids or bases or reabsorb acids or bases, getting my words confused, to balance pH of the blood. And one other thing it does is it converts vitamin D into its active form so that vitamin D can then be used for calcium absorption in the digestive tract. So you can see here the kidneys are doing a lot of things. And basically, if your kidneys shut down, you're unable to filter out waste products from the plasma. You also aren't able to balance pH, and it basically causes your blood to become toxic to your cells. So let's review the anatomy of the kidneys before we move into the more complicated physiology. So remember, if we look at a cross section of the kidneys, the outermost region is called the cortex, and then you have your renal pyramids, or they're called medullary pyramids. This area here, that's called the medulla. And then you have your renal pelvis. You had your minor and major calyxes that lead into the renal pelvis. Urine will enter into the renal pelvis and down the ureters to the bladder. Now, if we look at a microscopic picture of the kidneys, you'll see that histology-wise, they're made up of many of these components called nephrons. Okay, and we said that the nephron is the functional unit of the kidney because the nephron is where you produce filtrate. So the nephron has both a tubular component and a vascular component. And the function of the nephron is to filter the blood and produce urine. So let's first review the anatomy of the tubular component of the nephron. And remember, I'm going through this kind of fast and doing a general overview because this should be review from anatomy. Remember, you have the capsule that surrounds the glomerulus, which is a capillary bed. And this capsule is called the Bowman's capsule. You may have learned it as the glomerular capsule. I tend to call it Bowman's capsule. So what happens is blood moves into that capillary bed and fluid is forced out into the capsule. And when it gets forced out into the capsule, we now call that product filtrate. And the filtrate will travel through the nephron, tubular component. And the first part it travels to that comes off of the Bowman's capsule is the proximal convoluted tubule. And then that leads into this hairpin loop called the loop of Henle. From the loop of Henle, it enters into the distal convoluted tubule, that's here, and many distal convoluted tubules from different nephrons will feed into a larger collecting duct. And this collecting duct will run down through the medulla, through those renal pyramids, and the filtrate will drip into the renal pelvis. And once it's in the renal pelvis, that final product is called urine. Okay, it's called filtrate as it moves along because it's still getting adjusted. Things are getting added to it and things are getting reabsorbed as it moves through the nephron. And then the final product would be called your urine. Now let's review the vascular component. The kidneys receive a large amount of the blood supply. The kidneys will filter your full blood volume about 60 times per day. Okay, so there's a lot of blood moving through the kidneys. So remember your anatomy. You have your renal artery that feeds into the kidney, and then it will branch into the interlobule artery, and then you have the arcuate artery, and then you have these smaller interlobular arteries that then feed into afferent arterioles. So these afferent arterioles feed into a glomerulus, and then the glomerulus is drained by the efferent arteriole, leaves the glomerulus. And then it turns into another capillary bed that surrounds the nephron called the peritubular capillaries. Now in your longer nephrons that we call the juxtamedullary nephrons, it's basically their peritubular capillaries starting to straighten out and travel deeper into the medulla. And when they do that, they're called the vasa recta. So in your cortical nephrons, which are shorter, just in the cortex, we call them peritubular capillaries surrounding the loop. In juxtamedullary nephrons that extend deeper into the medulla, you have the vasa recta. I just mentioned this, but in case you don't quite remember the difference between a cortical and a juxtamedullary nephron. Cortical nephrons, the majority you can see here, if this is a cross section of the kidneys, you have the cortex here, this light brown color, the medulla, which is deep to it, in purple, and your cortical nephron is shorter, okay, and it's just a little bit of the loop extends down into the medulla. About 85% of your nephrons are cortical nephrons. About 15% of them are what we call juxtamedullary nephrons, where their glomerulus and capsule are closer to the medulla or deeper in the cortex, and then their loops are really long and they extend way down into the medulla. And these would be the guys that have the vasa recta. Alright, another region, anatomical region you need to remember from anatomy is the juxtaglomerular apparatus. So juxta means next to. So juxtaglomerular apparatus, it's these cells that are next to the glomerulus. So what we're looking at is if this is your nephron, there's your glomerulus and your Bowman's capsule. We're looking at this little region here. And what's interesting is you have your afferent arteriole that feeds in and your efferent arteriole that feeds out. Well, the distal convoluted tubule comes by and runs right back through that area. So if we look at this and we take a cross section of it and blow it up, we can see this picture over here in more detail. And so here's your capsule surrounding the glomerulus. You have your afferent arteriole feeding in. You have your efferent arteriole feeding out here. And then you're seeing a cross-section of the distal convoluted tubule that comes by there. So the juxtaglomerulary apparatus are these cells here, and there's different cells that we're going to learn about. But this area of the kidney or the nephron is responsible for regulating the rate that filtration occurs. So basically the volume that you produce per unit time of filtrate, and it also plays a role in regulating blood pressure. So the cells that do this is you have some columnar cells here that make up the wall of the distal convoluted tubule, and those are called the macula densa cells. They contain osmoreceptors, and their job is to regulate the flow rate of the filtrate, how fast it's going through the distal convoluted tubule. And the way that it detects how fast it's going is it detects the solute concentration. If things are moving really fast through the nephron, then you'd have more solutes left by the time it gets to the distal convoluted tubule because there wasn't time to reabsorb them. And so we know that the flow rate is too fast. If there's hardly any solutes left, you know that you had time to reabsorb it and maybe the flow rate is too slow. Okay, so it's detecting the flow rate. The granular cells are actually smooth muscle cells that you see here that are found in the wall of the afferent arteriole. And they have mechanoreceptors that are detecting stretch and pressure to monitor blood pressure. And these are the cells that release renin. So if blood pressure significantly drops in the afferent arteriole, that's a problem for the kidneys because the kidneys need pressure in the capillary bed to actually produce filtrate. So the idea is that the kidneys are concerned about blood pressure because it affects the rate of filtrate production. So if blood pressure significantly drops, then these granular cells can release renin and trigger the renin-angiotensin-aldosterone pathway. So let's start looking at some of the renal processes. There's 4 basic processes of the nephron. The first is glomerular filtration. So this is showing you a picture of, here's your afferent arteriole leading in, glomerulus, efferent leading out, and then the peritubular capillaries that follow along the nephron. So filtration is where you push fluid from the blood into the Bowman's capsule, and now that fluid is called filtrate. And then some of the components of the filtrate are going to be reabsorbed because you want to conserve them. And they'll be reabsorbed from the lumen of the nephron. Let me find my pointer. Lumen of the nephron into the peritubular capillaries, back into the blood. There's another thing that can happen is you can secrete things from the blood into the lumen of the nephron. Okay, and that's called tubular secretion. And then whatever's left, whatever wasn't reabsorbed or what was secreted, in the end will go into the renal pelvis and be considered urine. And so those things are excreted. So excreted means you're actually getting rid of them from the body. Secreted means it's going from the blood to the lumen of the nephron. Sometimes those terms get confused. So let's start with glomerular filtration. This is the first process. So what happens is plasma is filtered from the glomerulus into the Bowman's capsule. About 20% of the plasma that enters into the glomerulus actually gets pushed out into the capsule and becomes filtrate. About 80% of it just keeps going along the peritubular capillary and will have to get filtered next time it comes through the kidneys. Now, solutes and fluid can get pushed out to become filtrate. And we'll talk about how that happens. Things that are too large to get through the filtration pores can't get through and be part of the filtrate. So things that are too big to get through would be blood cells. Okay, so you shouldn't find in your filtrate or urine, you shouldn't be finding white blood cells or red blood cells unless there's some type of infection. And plasma proteins shouldn't be able to get through either. So in your urine, you should never be finding red blood cells, white blood cells, or protein. If you do find those things, that can be a sign of either inflammation of this filtration membrane which can be a sign of infection, which causes it to swell and allows more things to get through than normal. Or another thing that can cause it is hypertension, because if you actually have really high blood pressure and this glomerular pressure becomes really high, it can start to push things through that don't usually get through, such as proteins. So let's kind of look at the anatomy of this filtration membrane again. Remember, filtration is based on things moving from a higher pressure situation to a lower pressure situation. And it's non-selective. Everything gets pushed across. The only thing that's selected against is it's based on size. So it has to do with the size of the pores on that membrane, which determines what gets through. So if you look at this membrane, you have your glomerulus, which is a blood vessel. So you see the blood vessel here. And these blood vessels are fenestrated, which means that they are more permeable than other blood vessels or capillary beds elsewhere in your body. So they have little filtration pores. And then remember, you had podocytes. Do you remember learning that in anatomy? These podocytes are little cells that wrap around the capillary, the glomerulus, and they have little filtration slits also. So you can see they have little feet here that produce another filtration slit. I think of it as if you could take your fingers of your two hands and put them together. I can't really show you because you can't see me, but you know, if you take your right hand, your left hand, and you open up your fingers and you slide them together, it's kind of what a podocyte does. And so there's little spaces between where your fingers are. And if you look at a closer picture, you see here's the simple squamous cells of the blood vessel, and then they have a little membrane here, okay, basement membrane, and then you see the little feet of the podocytes on this side, and then over here is the Bowman's capsule, the lumen of the Bowman's capsule. So things get pushed from this higher pressure through the pores of the endothelium and then through the filtration slits. So there is sometimes, if you have infection, where you start to have swelling, and these cells, when they swell, the spaces get pushed further apart, and then things that usually don't get through, such as blood cells or proteins, might start to get through. Now, another reason why the glomerulus is really good at filtration, besides having pores, it is that the afferent arteriole is larger than your efferent arteriole. So what that creates is when more blood comes in than can get out, it creates a high pressure situation in this glomerulus. So this high pressure leads to filtration. So we can measure your glomerular filtration rate. So the filtration rate is the total amount of filtrate formed per minute by the kidneys. This can fluctuate depending on your body's needs, but the average glomerular filtration rate is you're producing about 125 milliliters of filtrate per minute. Now that's 180 liters a day. That is a ton. So your kidneys are producing 180 liters of filtrate. Now let me ask you this. Do you urinate 180 liters per day? No, obviously not. Okay. You probably produce about 1.5 liters of urine a day. So what does that mean? Well, what that means is you're producing a lot of filtrate that gets pushed into the nephron, but most of it is getting reabsorbed back into the plasma. Okay, and just a little bit remains as urine. So factors that govern how quick you produce filtrate. Okay. The main one, and this is what we're going to focus on, is the net filtration pressure, which we'll talk about. Other things that will affect it is the total surface area available for filtrate, so the number of nephrons. Okay, you have millions of them. You know, we're not going to talk much about this because there's not much to it, except if for some reason you have less surface area, such as less nephrons. One reason that could happen is if you are surviving on one kidney, which you can do. You can have one kidney removed and survive on one kidney. They've actually shown that if you do have one kidney, that kidney can actually produce more nephrons to help compensate. They have seen that. or if you have disease of the kidney that's somehow affecting the nephrons. The other thing is the permeability of the filtration membrane, and I talked about that already. Is if you have swelling of the membrane, it can make it more permeable and things will start to get through that shouldn't. The other thing is some autoimmune diseases, antibodies can get stuck in that membrane and kind of affect the permeability of the membrane. So mainly we're going to talk about net filtration pressure, because this is what your body can change in order to regulate the filtration rate. So how do you determine the net filtration pressure? Well, there's different pressures going on. And there's the glomerular capillary pressure, which is the blood pressure in the glomerulus basically. And that's about 55 millimeters of mercury. Then there's what is called the plasma colloid osmotic pressure. And this is because remember that things like to move from a higher concentration to a lower concentration. Basically, you retain proteins in the plasma because they can't get across to the lumen of the Bowman's capsule. So what will happen is the concentration of water is always going to be higher in the capsule than it is in the blood because the proteins stay in the blood and they're absent in the capsule. So there's an osmotic pressure. Water is going to want to tend to return back to the glomerulus by osmosis from the capsule. So that osmotic pressure is about 30 millimeters of mercury. And then there's what's called the hydrostatic pressure, is that once fluid moves into the Bowman's capsule, there is some pressure in the capsule. So what this looks like is you basically have to add up these pressures and see who wins. The blood pressure in the capillary, the glomerulus, is about 55 millimeters of mercury. So that's pushing fluid out. And then you have some back pressures, such as 30 millimeters of mercury for the osmotic pressure of water wanting to move by osmosis back into the glomerulus, and then just the pressure of fluid, the hydrostatic pressure of 15. So basically you have 55 minus 45 pushing this direction back into the glomerulus. So who wins? Well, 55 minus 45 is 10. So the hydrostatic pressure of the glomerulus is still 10 more millimeters of mercury. So it wins. So the net filtration pressure is still pushing fluid out of the blood and into the capsule. So that's what we call as the net filtration pressure. It's about 10 millimeters of mercury. Now this can change. If you have hypertension, this glomerular pressure could be even higher than 55. Let's say it's 65. Now you would have a net filtration pressure of 20. What would that mean? That would mean that you're producing filtrate probably faster. Now the kidneys are really concerned with regulating glomerular filtration rate. You want to keep it somewhat steady. And the reason you want to keep it steady is if you have uncontrolled shifts in the glomerular filtration rate, that means you can be producing filtrate too fast, and it could lead to fluid and electrolyte imbalances. Okay, so what will happen is if your glomerular filtration rate is too high, filtrate's going to move too quickly through the nephron, and you won't have time to reabsorb the things you need. To give you an example of things that you need to reabsorb, you reabsorb pretty much all of your glucose, all your amino acids, a lot of your sodium, a lot of your water. Obviously, right, if you're producing 180 liters of filtrate a day, but you're only urinating 1.5, that means you're reabsorbing, what is that, 178.5 liters you need to reabsorb. So if it's moving too fast through the lumen of the nephron, you won't have time to reabsorb it and will get lost in the urine. That could significantly affect your blood volume. If the glomerular filtration rate is too low, that means that the filtrate's moving very slowly through the nephron. Well, what can happen is everything gets reabsorbed. And this includes waste products like urea or your nitrogen-containing waste that you want to get rid of. If it sits there too long, you eventually reabsorb it. And that can be problematic. Point here, kidneys are concerned with controlling this glomerular filtration rate. You don't want it to be too fast. You don't want it to be too slow. So how is it going to keep that glomerular filtration rate consistent? To give you an idea, you might be asking, well, what's going to change my glomerular filtration rate? Well, one thing is, anytime your blood pressure fluctuates, that can affect glomerular filtration rate. So, for example, if your blood pressure increases because you're exercising, you may not necessarily want to increase your glomerular filtration rate. So the kidneys have a local control that even though your systemic blood pressure goes up, it can keep its glomerular blood pressure at its normal levels. Because the last thing you want to do when you're exercising is to start producing more filtrate and start losing fluid and become dehydrated. There's 3 mechanisms to controlling the glomerular filtration rate, and we're going to go in detail through each of these. One is you have some intrinsic controls, which I just mentioned. So the kidneys can regulate their glomerular pressure on their own, despite what the rest of the body is doing. Then there's extrinsic controls also. The sympathetic nervous system can regulate glomerular filtration pressure, and hormones can control it. This RAA stands for renin, angiotensin, aldosterone pathway or system. So we're going to go through these. Let's just look at this picture really quick so you understand what's going on. You have your afferent arteriole that feeds into the glomerulus here. If you increase glomerular pressure, that's going to increase the net filtration pressure, which increases your glomerular filtration rate. You're producing filtrate faster. So what happens is if arterial blood pressure goes up, that means glomerular pressure goes up, net filtration pressure goes up, glomerular filtration rate goes up. So what the kidneys will do is they'll want to bring your glomerular filtration rate back to normal. And one way it can do this is through renal autoregulation. So autoregulation means that factors within the kidney are regulating their glomerular filtration rate. And under normal conditions, autoregulation prevails. Okay, these local controls will prevail over sympathetic and hormones. Okay, under normal conditions. So if you look at this picture, if your blood pressure is anywhere between 80 and 180, your systolic blood pressure, your autoregulation in the kidneys is going to keep the glomerular filtration rate regular. So what we're saying here is your glomerular filtration rate is usually about 180 liters per day. So it's going to stay at 180 liters despite what's happening to your mean arterial pressure. Now what can happen though is if your blood pressure starts to significantly drop, autoregulation can't help at all. So let's say you start hemorrhaging or your blood pressure significantly drops, and we'll talk about why. And that's when, this part here, that's when now sympathetic nervous system is going to kick in. Now on the high side, you're kind of out of luck. So if you have severe hypertension, there's not really a mechanism to help there, and that's why hypertension can be a problem. Our body's evolved mostly dealing with normal fluctuations from things like standing, sitting, taking in more fluids, things like that, or being hurt, losing blood, certain things like that would happen that our sympathetic could deal with. We didn't necessarily evolve where we had chronic hypertension. That's a new thing due to our diet and lack of exercise. And so we don't really have a mechanism to deal with chronic hypertension where your blood pressure is constantly elevated. So let's first look at autoregulation. There's 2 types of autoregulation controls. There's the myogenic, and this involves, remember, muscle cells, and this is the smooth muscle that are found in the afferent arteriole, and they're going to respond to changes in pressure. So this afferent arteriole will detect changes in pressure and respond, and we'll talk about that. And then there's what's called the tubular glomerular feedback mechanism. And this is cells found in the macula densa that are going to detect the flow rate. And they're going to adjust the glomerular filtration pressure based on the flow rate. So let's look at each of these a little closer. Let's look at the myogenic mechanism. The granular cells and the smooth muscle cells found in this afferent arteriole can detect stretch. Okay. And what happens if pressure increases, then the afferent arteriole will constrict to lower glomerular filtration rate. Because the idea is if you increase pressure in the afferent arteriole, you're going to increase pressure in the glomerulus and increase filtration rate higher than you want it to be. So how we compensate for that, and this seems a little backwards because when you learned vasoconstriction, you learned increases blood pressure. Now when we're talking about the kidneys, you kind of gotta think backwards. So if I want to lower glomerular filtration pressure back to normal, the way I do it is I need to have less blood going into the capillary bed. So the way to get less blood to go into the capillary bed is to vasoconstrict this afferent arteriole. So if arterial pressure increases, the afferent arteriole will constrict. That will lower glomerular pressure back to normal. If arterial pressure decreases, let's say all of a sudden blood pressure drops in this afferent arteriole, that would cause your glomerular pressure to drop and your filtrate rate would drop. So what the afferent arteriole will do in that case, it will dilate. So if it dilates, that allows more blood into the capillary, and then capillary pressure will go up, so will net filtration pressure, and your glomerular filtration rate will increase back to its normal levels. Okay? So if blood pressure gets too high, then that means filtration rate would be too high. So you'd vasoconstrict, bringing the pressure back down to normal. If blood pressure dropped in the afferent arteriole, then your filtration rate would drop too low. So you vasodilate, getting more blood in there and bringing your filtration rate back to normal. Now the tubular glomerular feedback, okay, involves the cells of the juxtaglomerular apparatus, that's what JGA stands for, and it's involving your macula densa cells. So remember these macula densa cells here in the distal convoluted tubule will detect changes in flow rate and osmolarity. Now if they detect an increase in flow rate, if things are moving too fast through the tubule, remember that's dangerous. If it's moving too fast, you might be losing electrolytes that you need because you didn't have time to absorb them. Then these cells will release paracrine signals, vasoactive chemicals, that will cause the afferent arteriole to constrict. Because if I constrict this afferent arteriole, less blood's going into the glomerulus and it will slow down my filtration rate. If there's a decrease in flow rate, the macula densa cells will release chemicals that will cause vasodilation of the afferent arteriole. So in both mechanisms, you're controlling the flow of blood into the glomerulus, therefore controlling pressure in the glomerulus, therefore controlling the rate of filtration. So here's a little pattern of what's happening. Let's say my glomerular filtration rate it increases for some reason. So I have an increase in glomerular filtration rate. That means that the flow of fluid through my nephron will speed up. And if it speeds up too fast, that means I'm not reabsorbing all the electrolytes and solutes that I need to. So when it gets to this distal convoluted tubule, the macula densa cells detect that the flow has increased because there's more solutes in the fluid, and they'll release a paracrine signal that will diffuse to the afferent arteriole cells and cause the afferent arteriole cells to constrict. Okay, and when they constrict, that means that there's lower pressure in the glomerulus because less blood's flowing into the glomerulus, and your glomerular filtration rate will slow back down. Now, I kind of already mentioned this, so let's say you stand up, you sit down, your blood pressure shifts in this normal range here, but somewhere between 80 and 180, which is a huge range. Then autoregulation is going to control that. You're just going to change the diameter of your afferent arteriole to keep a constant glomerular filtration rate. Normally, autoregulation mechanisms prevail. But your afferent arteriole can only dilate so much if blood pressure drops. Okay, it can only get so big, right? So what happens is if blood pressure significantly drops, your autoregulation can't do anything about it. And what will happen is then your sympathetic nervous system will have to kick in. So this is when your sympathetic nervous system kicks in when blood pressure drops significantly. And this sympathetic nervous system can override the autoregulatory mechanisms, okay, to carry out long-term adjustments for blood pressure if blood volume drops. So what this means is your body has to kind of pick and choose what's most important. Most of the time you want to produce filtrate and get rid of your waste products. But if your blood volume and blood pressure significantly drop, what becomes your priority is conservation of fluids to maintain blood volume. So the last thing you want to be doing is producing filtrate. And when you kick into sympathetic, you're going to produce less urine because you're going to slow down your glomerular filtration rate, because that becomes your priority. If arterial blood pressure severely drops, then your baroreceptor reflex, that sympathetic reflex, will kick in and trigger vasoconstriction of your systemic arterioles. So all your blood vessels will constrict to increase your overall blood pressure. Now the afferent arterioles will also constrict by sympathetic, which will cause less blood flow through the glomerulus, and then therefore you'll decrease your glomerular filtration rate and reduce your urine volume. That's going to help conserve plasma volume and increase blood pressure. So when you're in sympathetic, you vasoconstrict that afferent arteriole and decrease urine production. Now, just to keep this clear, if you're talking about your autoregulation, when blood pressure drops, you want to vasodilate that afferent arteriole, right? To keep your glomerular filtration pressure normal. But if it gets too low, then sympathetic kicks in, you vasoconstrict, and you don't worry too much about urine production because you want to conserve blood volume and blood pressure. Alright, the other thing that's an extrinsic control is the renin-angiotensin-aldosterone mechanism. So the sympathetic nervous system can stimulate it, or it can be stimulated intrinsically. So there's a bunch of things that trigger renin release. One is an intrinsic control. When there's a drop in blood pressure in this afferent arteriole, there's less stretch to these smooth muscle cells, these granular cells. So when there's reduced stretch, the granular cells will release renin. Because remember, renin's released when blood pressure drops. The other thing is the macula densa cells can stimulate the granular cells to release renin. And the third thing is your sympathetic nervous system. So the granular cells here can be stimulated with epinephrine, norepinephrine, because they have beta-1 receptors. Remember your beta-1 adrenergic receptors. And that can stimulate them to release renin. And you should know this pathway. I think we've gone over it 2 times already. You learned it in endocrine, you learned it in cardiovascular, we talked about blood pressure, and here it is again. But let's go over it again. Your renin-angiotensin-aldosterone system. So granular cells in that afferent arteriole will release renin. Remember, renin will then convert angiotensinogen, which is an inactive protein in the plasma, into angiotensin 1. Angiotensin 1 gets converted into angiotensin 2 by, remember, the enzyme ACE, angiotensin converting enzyme. And then angiotensin 2 causes arterial vasoconstriction to increase blood pressure and also causes the adrenal cortex to release aldosterone. And remember, aldosterone leads to reabsorption of sodium and water follows. So that results in an increase in systemic blood pressure and an increase in blood volume. Alright, so that's the control of glomerular filtration rate. And this is a little summary of this, okay, how we control glomerular filtration rate. And I like this chart. It shows you down here, these 2 things are intrinsic controls. So you have your 2 intrinsic controls, which are the myogenic mechanism and the tubular glomerular mechanism. And then you have extrinsic controls here in green and yellow. Your extrinsic controls are hormones, the renin, angiotensin, aldosterone mechanism. Or you have neural controls, which is your sympathetic nervous system. Okay, and this is a really nice chart that shows all of this. So let's say your systemic blood pressure drops. Now, if it drops and it's somewhere between 80 and 180 millimeters of mercury for your mean arterial pressure, then your intrinsics are going to work. So that means blood pressure would drop in your afferent arterial, and that would cause the afferent arterial to dilate to bring your glomerular filtration rate back to normal. If systemic blood pressure drops, that would drop your glomerular filtration rate. Therefore, your flow rate would decrease, and the macula densa cells would detect that there's less solutes left in the filtrate because the filtrate's moving slower, and they'll release chemicals that will cause vasodilation of the afferent arterial. Now, if it significantly drops, then your priority becomes maintaining systemic blood pressure. So you can release renin, leads to renin-aldosterone pathway, increased sodium and water reabsorption, which increases blood volume, which increases blood pressure. Or you can have just vasoconstriction by the angiotensin II, which will increase systemic blood pressure. Or you can have just your sympathetic nervous system, which will cause the release of renin or cause the afferent arterial to constrict. So just keep that in mind. Under normal conditions, your intrinsic mechanism's priority is to keep your glomerular filtration rate at a normal rate. You don't want it too fast, you don't want it too slow. So despite small changes in blood pressure, they're going to keep a normal glomerular filtration rate. If blood pressure and volume significantly drop, then your priority becomes maintaining systemic blood pressure, and your extrinsic mechanisms like your sympathetic nervous system are going to kick in. Alright, so that's filtration. Glomerular filtration is pushing the fluid from the plasma into the capsule. Now, you already learned that most of the filtrate that originally gets pushed into the capsule and starts moving through the nephron, most of it's going to get reabsorbed. Okay, so you can see here about 20% of your plasma volume gets filtered out. 80% will continue on through the peritubular capillaries. And about 19% of that fluid gets reabsorbed back. So you can see here your reabsorption rates are high. So you filter about 125 milliliters of fluid per minute, and about 124 of that's going to be reabsorbed. About 99% of your water, 100% of all glucose will be reabsorbed that gets pushed out. As long as you don't have diabetes, and we'll talk about that. But in a normal individual, all glucose should be reabsorbed. You shouldn't be finding glucose in your urine. And then most of your sodium gets reabsorbed. Okay, so pretty much most things get reabsorbed. So let's talk about how these things get reabsorbed. Let's look at the histology of the nephron again. We'll review this from anatomy, but this is a picture. Here's your capsule. Here's your nephron. Now the nephron's lined by epithelial cells, okay, because this is the lumen of the nephron. And then you have interstitial fluid, and then you have your peritubular capillaries that are nearby. So your filtrate is here in the lumen of the nephron, and these are your epithelial cells that make up the wall of the nephron. So if you want to reabsorb something, it needs to move either through the spaces, but you have tight junctions. It basically needs to go through the epithelial cell, into the interstitial fluid, and then into the plasma. So we call this the luminal membrane here, which is the side of the epithelial cell that lines the lumen of the nephron. And then this is called the lateral space or basolateral membrane, which is on the basement membrane side where the interstitial fluid is. So let's look at sodium reabsorption first, because sodium reabsorption drives most of the reabsorption in the kidneys, and it's mostly through active transport. So what will happen is you have a lot of sodium-potassium pumps on these basolateral side of the epithelial cell. And so they're constantly pushing sodium into the interstitial fluid, and it will eventually move into the blood. So what that does is it keeps sodium levels low in the epithelial cell. It will always be higher in the filtrate. So sodium will move into the epithelial cell down its concentration gradient because it's always going to be lower here because you have a sodium-potassium pump on the other side. Now about 67% of sodium reabsorption happens in the proximal convoluted tubule. You also have some sodium reabsorption that's happening in the loop of Henle, and that's going to play a role in varying the concentrations of urine, and we'll talk about that at the end of this lecture. Now in the distal convoluted tubule, any of the sodium that's left, the reabsorption can be variable depending on your body's needs. And so that's going to be controlled by the hormone aldosterone. So sometimes your body might need to reabsorb more sodium or less, and that's going to be controlled by aldosterone secretion. So let's look at aldosterone. We learned about this in endocrine. He's going to work on the distal convoluted tubule, that's what DCT stands for, or in the collecting ducts. So pretty much in the proximal convoluted tubule in the loop, you're usually reabsorbing the same amount of sodium every time. Okay, it's kind of set, constant. Now, if your body needs to adjust based on its needs, that's going to happen in the distal convoluted tubule and the collecting ducts under the control of aldosterone. So how this works, let's look at the picture, is aldosterone will be released into the blood. So let's say, remember, aldosterone will be released for certain reasons. One is you are low on sodium. One is you're high on potassium. And the third reason is drop in blood pressure and blood volume. So you'd release aldosterone. It would travel in the blood to its target cells, which will be in the distal nephron. And aldosterone is a steroid, so it works through using an intracellular receptor. It will bind to the intracellular receptor. It will turn on the transcription and protein synthesis of certain proteins. And the proteins will lead to the insertion of new channels. So you have more sodium and potassium channels uploaded and more new sodium-potassium pumps on the basolateral side. Okay. So when you upload more of these channels, you can have additional sodium reabsorption in the distal convoluted tubule. And what also that leads to is, remember, the other function of aldosterone is to increase potassium secretion. And the way that this works is if you upload more sodium-potassium pumps here at the basolateral side, you're pumping sodium out, 3 sodium out for every 2 potassium in, and then they'll just follow their concentration gradient. Potassium will move into the lumen and be excreted, and sodium will move from the lumen into the blood and be reabsorbed. So let's go back to here. So about 8% of the filtered sodium is dependent on aldosterone. Okay, otherwise it will just be lost in the urine. Now, if aldosterone causes reabsorption of sodium, there's another hormone that will inhibit sodium reabsorption. We learned about this before, the atrial natriuretic peptide. And ANP is released from the atria when blood pressure is too high. So if blood pressure is too high, you want to lower blood pressure and blood volume. And one way you can lower blood pressure and blood volume is to inhibit sodium reabsorption. So how the ANP works is it acts directly on the collecting ducts to inhibit sodium reabsorption. The other thing that it will do is it will inhibit the renin, angiotensin, aldosterone pathway. So it will inhibit aldosterone production. And then it will also dilate the afferent arteriole, which will increase glomerular filtration rate, which will reduce water and sodium reabsorption, because if you get things moving through the lumen faster, you don't have time to reabsorb as much. Okay, so it kind of works through a few mechanisms on the kidneys. One is it works directly on the afferent arteriole, it works directly on the collecting ducts, and it inhibits the renin-angiotensin-aldosterone pathway. And here's just a picture of that, what the ANP is inhibiting. It's inhibiting sodium reabsorption at the tubules, it's inhibiting the renin-angiotensin-aldosterone, it's inhibiting the smooth muscle of the afferent arteriole, which causes dilation, which speeds up filtration. And it actually also inhibits the sympathetic nervous system, so that will decrease arterial blood pressure. So that's reabsorption of sodium. Let's look at the reabsorption of some of the other things that your kidneys will reabsorb. One is you reabsorb most of the water. Remember, 124 out of 125 milliliters of fluid per minute. You're going to reabsorb all your glucose, most of your amino acids, and some of your anions like chloride and bicarbonate. So most of these are all linked to the active reabsorption of sodium. So by actively pumping sodium, using your sodium-potassium pumps, it creates certain gradients. So if you're reabsorbing sodium, so on this picture, if sodium's moving because of sodium-potassium pumps actively, then passively, water's going to follow, right? Because water's going to follow by osmosis. It's going to follow the solutes. And water moves through pores called aquaporins that can be inserted into the epithelial cells. Anions are going to also follow by diffusion down their electrochemical gradient because sodium's positive, it's a positive ion, and so the negative ions, your anions like chloride, will just follow it. And then glucose and amino acids move by secondary active transport. Now remember, secondary active transport relies on primary active transport, which is your pumping of your sodium. So you can see here, if sodium's getting pumped out using ATP, requires energy, you're going to have a higher concentration of sodium here than you do in the cell, than you do in the lumen. And water's just going to follow that gradient by osmosis. So it will move through an aquaporine, and then it will move into the interstitial fluid, and as pressure increases here, it will move into the peritubular capillary. Now when we look at water reabsorption, 80% of it is required. You're obligated to reabsorb it just to maintain blood volume. And most of that's going to happen in the proximal tubule and the loop of Henle. And it's going to occur by osmosis. The rest of the water is facultative, which means it can be adjusted. Okay, you're not required to reabsorb it. And that's going to happen in the distal convoluted tubule. So it's going to be based on your body's needs. If you need to reabsorb additional water, you can. If you don't, then you will excrete it. So this occurs in the distal convoluted tubule and the collecting duct, and it's under the control of your antidiuretic hormone. So you've learned about ADH before. Another name for ADH, remember, is vasopressin. See how everything you've learned is now coming back and coming together here at the end? So vasopressin will be released from the pituitary gland, and that's in response to changes in osmolarity in the blood. And it will travel in the blood and will bind to a receptor on the distal convoluted tubule cells or the collecting duct cells, and through, guess what, a secondary messenger, your cyclic AMP pathway, it will cause the protein synthesis of more aquaporins, which are protein channels, that it will be inserted in the lumen side of your epithelial cell, and that will speed up water reabsorption in those distal convoluted tubules. So it increases water reabsorption and it produces a more concentrated urine because there's less water that gets excreted, more solutes. And then lastly, let's look at glucose and amino acid reabsorption. Now this involves secondary active transport. You learned about this back in our first unit, and it gets co-transported with sodium. It's similar to what happens in the digestive tract, how you absorb in the small intestines your glucose and amino acids. Okay, so remember on the basement membrane side of the epithelial cell, you have a sodium-potassium pump. That's your primary active transport, and it's pumping sodium out using ATP against its concentration gradient. So sodium's always high in the interstitial fluid. It's going to be high in the lumen, and it's going to be low in the epithelial cell. So what that creates is a sodium gradient. So sodium is going to want to move from its high concentration to its low concentration. And you have a glucose cotransporter. That glucose will move with the sodium using its gradient to move into the epithelial cell. So it's moving against its concentration gradient, because if your glucose is lower here and it's higher in the cell, you will still reabsorb that glucose because it's riding on the sodium gradient. So even though this transporter doesn't directly require ATP, it indirectly requires it because it requires you to create a sodium gradient, and that's called secondary active transport. Now, amino acids will be reabsorbed the same way. So I told you you should not find glucose in your urine if you're a healthy individual. If you do find glucose in your urine, what that could mean is that you reached your glucose transport maximum. Basically, your kidneys can only reabsorb so much glucose. It has its limits. We tend to look at this for glucose, but you could look at this. Any solute that you reabsorb has a transport maximum that you can calculate. And the idea is why there's a maximum is there's only a certain number of carriers, for example, your sodium glucose carriers, in the renal tubules available. And they can only work so fast to get the glucose across the membrane. So what happens is you can saturate those transporters. And once they're saturated, anything that doesn't get pumped across or moved across will be found in the urine because it will be excreted. So if you look at glucose, for example, it depends on your blood glucose levels. As long as your blood glucose levels are normal, or even if they're high, and your kidneys can usually still reabsorb most of it. So what we're looking at here is your plasma concentration, your blood glucose levels. 80 to 100 is normal. Okay, about 90. Above that would be considered high, but when you get to about 400, that's considered critical. Your glomerular filtration rate, you filter about 125 milliliters per minute, and this is showing you about the amount of glucose that's going to get filtered per minute. So the kidneys, the channels can only move about 375 milligrams of glucose across per minute. So if you have about 100, you're going to be able to reabsorb all of that. So none of it would be excreted. But eventually, if you start to get up to 400, okay, and you filter about 500 milligrams of glucose per minute, and you can only move 375 milligrams per minute across, that means you're going to be left with some in the filtrate, 125 milligrams, or whatever you have over 375, which is your transport maximum, and that's going to be excreted and found in the urine. So finding glucose in the urine is a sign of diabetes or elevated blood glucose levels. Alright, so I told you reabsorb most of your water, most of your sodium, all your glucose, most of your amino acids, a lot of your anions, but substances that are not reabsorbed are usually your nitrogenous waste. Some of it is reabsorbed, but what you would find in your urine are your nitrogenous wastes, such as urea, creatinine, uric acid, ammonia, and other nitrogens. The reason they aren't reabsorbed is they usually lack carriers. There's not channels for them to get reabsorbed. They don't need a channel if they're lipid soluble. Another reason that they're not absorbed is they lack channels and they're not lipid soluble, or they're too large to pass through the membrane pores. So those are reasons why substances would not be reabsorbed. Urea, you find about 50% of urea will get reabsorbed, and that's because it is lipid soluble. And they originally thought that there wasn't any carriers for urea, and they've actually found that there is some. But you're only reabsorbing about 50% of it. And all your other waste products get excreted. Alright, so let's look at our third process. So we've talked about how you produce your filtrate, how it gets pushed into the lumen, and then how you reabsorb things back into the peritubular capillaries. And now we're going to talk about tubular secretion. It's a selective process, and you're moving things from the peritubular capillaries from the blood into the lumen of the nephron. Why would you now want to move things into the lumen? You already filtered it, and then why would you move things here? Now the reason is it provides a mechanism to speed up the elimination of substances from the blood. So remember I told you that you only filter about 20% of the plasma originally here up at the glomerulus, and 80% of it keeps moving. Well, guess what? You'd have to wait for that 80% to come back to the kidneys again to get it filtered. So if there's something in the blood that you want to get rid of now, you don't want to wait for it to have to come back to the kidneys, you could use tubular secretion to actually get it into the filtrate this time around. So it's important for disposing of substances that aren't already in the filtrate. It's good for eliminating undesirable substances that could be toxic, like urea or uric acid, your nitrogen-containing waste. And the two other things that it's very important for is these two things. Your body's very sensitive to potassium levels. It can affect your cardiac muscle tissue. It can also affect skeletal muscle, but you're really worried about your cardiac muscle tissue. So if you have high potassium levels, you need to get that potassium excreted immediately, and then also controlling blood pH. So let's first look at the secretion of potassium. Potassium is almost completely reabsorbed in the proximal convoluted tubule. So if you then have high potassium levels, you're going to need to secrete it in the distal convoluted tubule. And this is going to be under the control of aldosterone. And we've already kind of learned about this. Aldosterone will cause more sodium-potassium pumps to be inserted. And so you're pumping more potassium into the epithelial cell from the interstitial fluid, and then it will move down its concentration gradient into the lumen. Aldosterone will be stimulated to be produced if you have an increase in plasma potassium levels, and that would increase potassium secretion, and therefore you'd have higher amounts of potassium in your urine. The other thing is if you have low sodium or low blood pressure, so through the renin-angiotensin-aldosterone pathway, that would cause increased sodium reabsorption. Now the other thing is secretion is used for acid-base balance. You've already learned how your body maintains its pH, and pH is important because acidity can denature proteins and all your enzymes that you need to function. You also learn that a lot of your enzymes work at an optimum pH, so if it becomes too basic or too acidic, your chemical reactions will be affected. So it's very important to maintain pH homeostasis. And the way that you regulate the concentration of hydrogen ions, which is how you measure pH, we learned that you have chemical buffers that are in your blood, and those react within seconds. Now if those become overwhelmed, then your respiratory system will kick in, and you've learned that you can regulate pH by changing your ventilation. You can increase your ventilation rate to get rid of carbon dioxide and therefore cause your blood to shift from acidic to more basic, or the other way around. If you're somewhat alkalosis, you can decrease ventilation rate, and then that's going to kick in within a minute or so. And then the other way you can regulate acids and bases is through the renal mechanism, or your kidneys. Your kidneys are going to take a little longer to actually make a change in the blood pH, hours to days. So the general idea here, and this makes complete sense, if you are in acidosis, you have an increase in plasma hydrogen ions, or you can have an increase in carbon dioxide, because we learned that that will lead to increased hydrogen ions. So what would your kidneys want to do? Well, if you have an increase in hydrogen ion concentration, then you're going to want to increase hydrogen ion secretion and get rid of them. excrete those extra hydrogen ions in the urine. Or, you're kind of doing both, increase your bicarbonate conservation, because bicarbonate acts as a base by binding hydrogen ions. Okay, so I want to conserve it, and you'll find less bicarbonate in your urine. Okay, and that's going to alleviate your acidosis. So what are the renal mechanisms that are used for regulating acid-base balance? The main thing is you're usually messing with bicarbonate ions. You can either conserve, which means you're reabsorbing all the bicarbonate ions that you filtrate, or you filter, and you can then generate new bicarbonate ions. So if you're in acidosis, you're going to conserve, reabsorb all your bicarbonate, and you can generate new bicarbonate ions to counteract additional hydrogen ions. If you're in alkalosis, you can excrete your bicarbonate ions, conserve your hydrogen ions. So the idea is, you kind of got to think, losing a bicarbonate ion is somewhat the same as gaining a hydrogen ion, because it's now not there to buffer the hydrogen ion in the blood. And reabsorbing a bicarbonate ion is kind of the same as losing a hydrogen ion, because if you have the bicarbonate ion, you now can have it bind to the hydrogen ion. And this little chart kind of summarizes it. If you're in acidosis, if your blood is acidic, you're going to secrete and excrete hydrogen ions, conserve and reabsorb bicarbonate. So you shouldn't find any bicarbonate in the urine, okay, because you want to keep it, and your urine will be somewhat acidic because of the secretion of the hydrogen ions. If you're in alkalosis, okay, you want to conserve hydrogen ions so you don't excrete them, and you don't want to reabsorb or make any new bicarbonate to add to the plasma because you're in alkalosis. So you'll find a large amount of bicarbonate in the urine, and your pH of your urine should be somewhat alkaline. So let's look at how you carry this out. For the most part, your kidneys will reabsorb all the bicarbonate that it secretes, because those bicarbonate ions are necessary to buffer hydrogen ions in your blood. Okay, because bicarbonate is the buffer system. So how you reabsorb the bicarbonate that you filtrate is filter is kind of an indirect mechanism, because there isn't bicarbonate channels in the epithelial cells of the proximal convoluted tubule, but you still reabsorb it. And the way you reabsorb it is by converting carbon dioxide, because carbon dioxide is lipid soluble and can move into the epithelial cell without having a channel. So that's why this is kind of backwards, but this is what happens. You filter bicarbonate, okay, and it will bind with a hydrogen ion to form carbonic acid, which will dissociate into water and carbon dioxide. Now this carbon dioxide can diffuse into the epithelial cell, and in the epithelial cell you have the enzyme, remember, carbonic anhydrase, which is the enzyme that then causes carbon dioxide and water to form bicarbonate, and then it will dissociate into hydrogen ions and bicarbonate. And in this hydrogen ion, there is a hydrogen pump that will pump it out, and the bicarbonate will get reabsorbed. So see how it's kind of a really indirect way? You can't just directly take this bicarbonate and reabsorb it. You don't have bicarbonate channels, but you do have hydrogen ion channels that pump the hydrogen ion out, that binds to the filtered bicarbonate, which produces carbon dioxide. That carbon dioxide goes into the cell and gets turned back into bicarbonate, and then the bicarbonate gets reabsorbed. In general, bicarbonate is disappearing from your filtrate at the same rate it's getting reabsorbed into the blood. So you reabsorb all your bicarbonate that you filter. You have bicarbonate in your blood because it's the buffer. And what it's buffering is a lot of the acids that your body produces. You produce acids from metabolism. Some of your amino acids are acidic. From carbon dioxide, that's a product of cellular respiration. Okay. So your body's continually, just from metabolism, producing acids. And that's the function of the bicarbonate in the blood, to buffer those acids. So let's say you just have, I'm just throwing a number to get you to picture this. Let's say you have 100 bicarbonate ions floating in blood. You have a lot more. You have millions of, but let's say you just have 100. Those 100 get filtered out of the blood into the lumen. Well, guess what? You gotta reabsorb all those bicarbonate because their job is to buffer the acids you produce from metabolism. So that system's in balance, let's say. You have enough bicarbonate to buffer your metabolism. But then something changes. For example, your diet brings in more acid, hydrogen ions. Well, now you're going to need to produce new bicarbonate to deal with these additional hydrogen ions. Or let's say you start exercising and produce lactic acids. You're going to need to produce additional bicarbonate to counteract these additional acids. Does that make sense to you? So you have a certain amount of bicarbonate that can deal with your typical acids that are produced with metabolism, and you're going to reabsorb all of those in the kidneys to keep them in the blood. But if you have additional acids that get added because of diet or other circumstances, and you go into acidosis, then you're going to need to generate new bicarbonate. So two mechanisms where you can generate new additional bicarbonate ions to buffer additional acids. One, you're going to excrete acids, so you're going to excrete hydrogen ions, and you're going to make new bicarbonate ions to reabsorb. Or the other thing you excreted as is ammonia, and we'll talk about this. Your pH of your urine is going to become pretty acidic. And your pH of your urine can be very acidic, about 4.5. But if it was to get any more acidic, it can be dangerous. So hydrogen ions usually have to bind to buffers that are in your urine also. So that could be amino acids that can buffer it, or phosphate buffer systems. Let's look at how you deal with acidosis of the blood. So the kidneys are going to generate bicarbonate ions and add them to the blood. And at the same time you're adding a bicarbonate to the blood, a new one, you're going to excrete a hydrogen ion into the lumen. And the hydrogen ion is going to bind to a phosphate buffer so that it doesn't cause your urine to become too acidic. And the way that your kidneys generate these new bicarbonate is they use carbon dioxide. So basically carbon dioxide and water bind to form carbonic acid, which is going to dissociate into hydrogen and bicarbonate. And this new bicarbonate is going to be reabsorbed. Another way you can produce new bicarbonate is to actually use amino acids. So the amino acid, for example, glutamine, can be converted into or metabolized to produce ammonium, 2 ammonium molecules, and 2 bicarbonates. And these bicarbonates, these 2 bicarbonates, are going to be reabsorbed, or not reabsorbed because they're new. They're going to be absorbed into the blood, and then the ammonium is going to be excreted. Actually, what happens is you produce ammonium, which then binds with hydrogen to become ammonia, and you're going to excrete it. 2 ways you can produce new bicarbonate is from carbon dioxide or from amino acids. But in any case, you're reabsorbing the bicarbonate, you're excreting the hydrogen. The hydrogen is either getting excreted directly into the lumen and binding to a phosphate buffer, or it can bind to produce ammonium. Now what if you're in alkalosis? And in this case, type B cells, I should go back. These cells here that deal with acidosis are called type A cells. So think A, acidosis. Type B cells are a different type of cell found in the nephron. And think B, basic, which stands for alkalosis. Okay, when you're alkalosis. So what they do is they secrete bicarbonate and reabsorb hydrogen ions. And just all it has to do with these different types of cells have different channels. So, and on what side the channels are on. So on these type B cells, your proton pump is on the basolateral side, and it's when you produce bicarbonate and hydrogen ions from water and carbon dioxide. In the type B cells, the hydrogen ions get pumped into the basolateral side and into the blood, and the bicarbonate gets pumped using your chloride bicarbonate pump into the lumen. Okay, and this is the same kind of pump that's found in your red blood cells for the chloride shift. So does that make sense? If you go back here, the channels are just on the opposite sides in your type A cells. Your proton pump is on the lumen side, and your bicarbonate pump is on the basolateral side. Now the kidneys have a remarkable way of being able to make either really dilute large volumes of urine or very small volumes that are really concentrated. And this depends on the body's needs. If you need to retain water and conserve it, then you would produce a small volume of urine that's really highly concentrated in solutes. If you have a lot of water that you need to excrete, you can produce a large volume of urine that's dilute with solutes. So the kidneys can produce urine in a wide range. It can produce a little volume from 0.3 milliliters per minute, which is a very small volume. That could be about 1200 milliosmoles per liter. That's very, very concentrated. The osmolarity of your blood is about 300 milliosmoles. So you can make it very hypertonic compared to your blood and interstitial fluid. 2, producing a really large volume, 25 mils per minute, which is a lot, at about 100 milliosmoles. So urine that is hypotonic to your blood and interstitial fluid. So how is the kidneys able to do this? The way it can produce urine that's more concentrated than the actual body fluids has to do with that there's a vertical osmotic gradient found in the interstitial fluid of the kidneys. So when you look at the kidneys, the cortex, the interstitial fluid surrounding the nephron and the peritubular capillaries, the osmolarity is about 300 milliosmoles, which is isotonic to your blood and interstitial fluid. And as you go down towards the renal pelvis in the medulla, you can see that it gets more concentrated or hypertonic. It goes to 600, 900, and then 1200 milliosmoles is the most concentrated. Now, this concentration gradient follows the anatomical structures like your loop of Henle, how it runs down through the medulla, and the collecting ducts, remember, run down through the medulla towards the renal pelvis. And how is the kidneys able to have this osmotic gradient? Because remember your laws of diffusion. You would think that you wouldn't be able to establish an osmotic gradient that is hypertonic to your body fluids, because the way solutes move is they're always moving from a higher concentration to a lower concentration. So you would think the solutes in water would just move until everything evens out. So how is your kidneys able to establish this gradient? Well, it has to do with something called the countercurrent system. Any countercurrent system means that you have things moving in opposite directions. And in your kidneys, your filtrate moves through the loop of Henle. The descending limb of the loop runs in opposite direction of the ascending loop. So you have filtrate moving in opposite directions of each other, very close to each other. And this also has to do with the vasa recta, which is the blood vessels that are in association with the nephron. So the vasa recta also runs in a countercurrent system to each other and to the loop. So you could see that the descending loop runs countercurrent to the ascending loop of the vasa recta, and the ascending loop of the loop of Henle is running countercurrent to the descending vessel of the vasa recta. Okay, so this is what countercurrent means. So we'll talk in a minute about how this allows you to keep your gradient. But we call this specifically the countercurrent multiplier, because it refers to the ability to increase the osmolarity of the interstitial fluid as you travel down into the medulla. And it has to do with the two properties of the limb, of the loop. So the descending limb of the loop of Henle is impermeable to solutes, but is permeable to water. And the ascending loop is permeable to solutes but impermeable to water. So as filtrate is moving down through the through the descending limb here, water gets reabsorbed. And so the filtrate ends up being about 1200 milliosmoles at the tip of the loop. And as it comes back up, water can't move back in, but ions can move out. So you can start to have ions move out and decrease your concentration when it gets back up into the cortex. If you have a gradient, so over here it's showing you the gradient as you travel down into the medulla. So this is the interstitial fluid surrounding the nephron. So it's about 300 in the cortex. I have to apologize. I know when you guys see my lecture, you can see the arrow highlighted moving around frantically, but I can't see it when I'm doing the lecture. It's not highlighted for me. It's just a white arrow. So I lose it all the time, and that's why you see it shaking around on the screen, because I have to move it around to try to find it. So I apologize for that. I'm not just frantic with my mouse. I'm just trying to find the pointer. But you can see as you move down in the medulla, it goes up to about 1200 milliosmoles. So it's really concentrated down here. So what happens is, based on the laws of diffusion, so the filtrate is 300 milliosmoles. So as it starts to move down, your filtrate is hypotonic to the interstitial fluid. So water is going to move from where it's a higher concentration to its lower concentration. So water will start to move out, okay, because it's going to be in a lower concentration as it gets more concentrated as you get down into the medulla. Okay, so does that make sense? So if you're 900 milliosmoles here, but it's 1200 in the interstitial fluid, water is going to move out to its lower concentration. Because remember, the higher the milliosmoles, that's the amount of solutes. So if you have 1200 milliosmoles of solute, that means you have less water than if you have 900 milliosmoles of solute, then you have more water. So water is moving out. And what that creates is water will move out until the filtrate goes to equilibrium with the interstitial fluid. Okay, so the filtrate ends up being about 1200 down here, same as the interstitial fluid. Now you would think that then also salts could move in, and so until everything goes to equilibrium, but they can't because this descending limb is impermeable to salts. And as you go up, water can't move back in as it would want to because this limb is impermeable to water but permeable to solutes or salts. And so sodium chloride is going to be actively transported out as you travel up. And that's what keeps your gradient. Because if water was just moving out, it would dilute the concentration gradient of the interstitial fluid. So to keep the concentration gradient, you would have to pump sodium chloride out to counteract that water. And the reason it's important to keep that concentration gradient there is not necessarily here for the loop, but later on when filtrate moves down the collecting duct. So the loop helps maintain the osmotic gradient so that later on when filtrate moves down through the collecting duct, you can adjust it. Now, I mentioned that the vasa recta also runs countercurrent, and we call this the countercurrent exchanger. So the loop is responsible for maintaining the gradient, but so is the vasa recta, because what it's doing is, as water moves out, it can get reabsorbed by the vasa recta. So you're going to move salts out, and they can move into the vasa recta because the vasa recta is running countercurrent. And then water from the descending loop will move into the vasa recta and dilute it. So what's happening, and this is important for the vasa recta, is it has to run countercurrent because it's 300 milliosmoles your blood when it comes in. And you need your blood to also be 300 milliosmoles when it leaves. Otherwise, you would significantly affect your osmolarity of your blood. Because if the vasa recta only ran in one direction and not countercurrent, let's say it came in at the cortex and it left at the medulla and it didn't run countercurrent and come back up, what would happen is it would go to equilibrium with the interstitial fluid. The blood coming in would come in at 300. As you get more concentrated, interstitial fluid surrounding the blood vessel gets more concentrated. Water and salt would move in and out until it went to equilibrium with the interstitial fluid. So the blood leaving would also be 1200 milliosmoles, okay, equal to the interstitial fluid. That would not be good. Okay, so you have this countercurrent system, so that blood leaving is also 300 milliosmoles, the same as when it came in. And then this exchange here with the countercurrent multiplier, it's just creating this osmotic gradient where you have it going from 600 to 1200 milliosmoles. And this is where the purpose comes in. Okay, so now you'll see why we had to have this gradient. It comes into play when your body wants to produce really concentrated urine. And what's going to control that is the antidiuretic hormone. Because if you want to produce very concentrated urine, you're going to reabsorb your water and leave the solutes. So you've created this osmotic gradient, and then the filtrate is traveling in the distal convoluted tubule and will come to the collecting duct. Now, if ADH hasn't been released, the collecting duct is impermeable to water. So what will happen is whatever the osmolarity is of your filtrate, when it gets to the collecting duct, it will stay that osmolarity because the collecting duct's impermeable, and it will just leave. The urine will be about 100 milliosmoles, pretty dilute. But if you want to make it more concentrated, you release ADH, and ADH will make this collecting duct permeable to water. The filtrate is hypotonic to the interstitial fluid. So as it moves down, water is going to move out towards its lower concentration gradient. Because as the interstitial fluid gets more concentrated, more and more water is going to move out until you can get to, if you make that duct very permeable, your urine can get to about 1200 milliosmoles because that's the highest concentration of the interstitial fluid. So that's the whole purpose of the loop, is to create the osmotic gradient and maintain that osmotic gradient. And you keep that osmotic gradient so that if you want to produce really concentrated urine, you can make this collecting duct permeable to water by releasing ADH, and then the water will get reabsorbed and you can produce really concentrated urine. If you don't want to reabsorb the water, then you have an absence of ADH, and the filtrate will just stay at the osmolarity of 100 milliosmoles as it travels down through the collecting duct. Now, there is some things called diuretics, which are things that cause you to produce large volumes of urine. Okay. So osmotic diuretics, there's a few. One thing is high glucose levels in the filtrate. So if you're a diabetic and you have high glucose that's still in the filtrate because you've reached your transport maximum, it will carry water out with it, because now you'll have a higher filtrate, will be more hypertonic, and the water will stay with the solutes. So people that are diabetic, a lot of times they produce a high volume of urine, and then they're always thirsty. Another thing is alcohol can be a diuretic, and the way alcohol is a diuretic is it actually inhibits the release of ADH. Caffeine can be a diuretic because it inhibits sodium ion reabsorption, and then there's a ton of drugs that are diuretics that you can obviously take. And most of the diuretics, such as Lasix, they inhibit sodium reabsorption. They can work on different sodium channels because there's different types of sodium channels in the kidneys depending on what drug it is. But in the most part, they're working on some of the sodium channels to inhibit their reabsorption. And so then therefore the water won't follow. It will stay in the filtrate and you'll produce a larger volume of water. For hypertension, using diuretics is also a treatment because you can get rid of volume. So if you decrease blood volume, you can help control blood pressure. We've talked all about how you produce the filtrate from the blood and then the filtrate travels through the nephron. You reabsorb what you need, you secrete what you don't need, so you've adjusted the filtrate based on your body's needs, and whatever is left when it gets to the collecting duct, then you're going to excrete that. Okay, so your excretion is the elimination of whatever remains in that tubular lumen, and we call that final product urine. And so urine is going to enter into the renal pelvis and then into the ureters that lead from each kidney, and the urine will be transported to the bladder, and it travels through the ureters, one based on gravity but also peristaltic waves because the ureters have smooth muscle in them, and then it will travel to the bladder, and the bladder's job is just to temporarily store the urine. And the bladder's smooth muscle, and it will start to stretch as it fills. And there's a reflex called the micturition reflex that is basically the reflex of emptying your bladder. So what will happen is, as the bladder fills, okay, the bladder sits right behind your pubic bone, and as it fills, it fills up into the abdominal cavity. And as it fills, it will activate stretch receptors in the wall of the bladder, and that's what triggers the reflex that causes the smooth muscle of the bladder to contract, and then the internal sphincter to relax. And urine will be passed through the internal sphincter. But you also have an external sphincter, which is actually skeletal muscle that you can control. So you have to voluntarily relax that external sphincter. So this is what the reflex kind of looks like visually. You have your bladder, and then you have a neuron that travels to your external sphincter, which is skeletal muscle, that causes it to stay contracted, and you can voluntarily control that. Now your internal sphincter is smooth muscle, and it is involuntary. And so your bladder fills, and as it fills, it triggers stretch receptors, and that impulse gets carried on a sensory neuron to the central nervous system, and then out through. It's a parasympathetic reflex because you're controlling smooth muscle, so it's autonomic. And so parasympathetic neuron will cause that smooth muscle of the bladder to contract and the internal sphincter to relax. Now you also have to then voluntarily relax your external sphincter, so you can control that. But that's a learned reflex. Like babies haven't learned to control their external sphincter yet. Okay, but as you get older, you start to learn that. Now what's interesting about the bladder is, as the bladder fills, it says it will fill to about 200 milliliters, and that will trigger your stretch receptors. So you become aware of, okay, I need to go to the restroom. But what can happen is, if you don't go to the restroom right then, have you ever had the experience where then you forget you have to go to the bathroom for a little bit, and then about 20 minutes later you're like, oh yeah, I gotta go to the bathroom. But now you have to go more. Okay, and so what's happening there is it will tell you, that your bladder is full, but if you don't choose to voluntarily go to the restroom, the smooth muscle will adapt. Remember we talked about how smooth muscle has a stretch, relax ability. So it will stretch and it will kind of adapt until it fills a little more. So then it will fill another 200 mils, and then it will remind you, okay, I'm getting fuller. Eventually, you can't ignore it anymore, and they say that your bladder can hold about 500 milliliters, but that depends on person to person.